Hi everybody, it's time to talk about antibiotic resistance. So first, a really short history of antibiotics. Everybody knows that penicillin was discovered by Alexander Fleming, and it's often told as a stereotypical example of a scientific discovery by accident, the holy cow moment. Scientific discoveries happen by pure chance. So a slightly more complicated version of that story is that it's true that the discovery that the fungi on his Petri plates were an accidental contaminant from the window were producing an antibiotic that killed the bacteria on the Petri plate. So the part about the contaminant, I think, is roughly true. The part that the very short version of the story leaves out is the fact that Fleming had dedicated his career to finding a molecule that kills bacteria. Right, so he had been working his whole life, basically, or at least for decades, of trying to find a good chemical that would be useful for treating bacterial infections. And so when he found something that was clearly killing his bacteria on a Petri plate, he was very prepared to take advantage of that observation, follow up on it, and convert it into an actual drug that was useful. Right, so that's a more complicated and I think more important version of that story. Another aspect of antibiotics that people tend to forget is that they're really recent. So Fleming's observation was 1928, so we're clearly in the 20th century. And the first time they're really used widely as a useful drug was actually during World War II. I've seen estimates that the use of antibiotics in the field during World War II might have been one of the biggest advantages that the Allies had over the Axis powers. And even though this is a very recent invention, basically as soon as the first antibiotic was used widely, resistance to that antibiotic was already noticed. So this is a technology that has to be constantly renewed, and that is why we are talking about it today. It's very much a very important topic today in medicine and in our everyday lives. So I've seen one estimate that there have been at least 2 million illnesses and 23,000 deaths that can be attributed to antibiotic resistance, and depending on how you do those calculations, it could be a lot larger. And one theme of what I'm going to teach you today is that one reason why this is a medically challenging problem is because antibiotic resistance can be spread between bacterial species by transferring genes. So it does not rely on the traditional Darwinian descent of genes from parent to offspring. Bacteria can share the gene among completely unrelated species, and that is one of the very important reasons why this is such a tough problem. Another aspect of this that is relevant to this course is that the research going on now to find new antibiotics, which is a constant ongoing search and will probably always be something that humans are doing, because antibiotics have inherently limited effectiveness, as we're going to talk about soon. This research to discover new antibiotics, it's inherently a research program centered on exploring microbial diversity. In order to find new antibiotics, we have to discover new species of microbes, and to understand how they work and to understand how they live in their natural habitats, because antibiotics are a product of natural microbial ecology. Microbes have been producing chemicals to kill each other ever since there have been microbes, so for billions of years. And humans only recently got involved in this game, right? So understanding microbial evolution, microbial ecology, microbial physiology, microbial diversity are absolutely essential for making any kind of real progress on this problem. Okay, that's kind of my short spiel on this. I'll get off my soapbox a little bit and I'll just talk about the biology. This is a really cool experiment done a few years ago. To demonstrate how quickly antibiotic resistance can evolve, and I use the word evolve very explicitly. That's exactly what this experiment was designed to detect and to show, actual biological evolution occurring in real time. So the way they did this is that they constructed a gigantic rectangular Petri plate. So this thing is over 1 meter long, so it's over 3 feet long, and on each side of it, they inoculated some E. coli. So just like a normal Petri plate, they put some E. coli on this side and on this side. And then in different sectors, different regions of this rectangular plate, they had different concentrations of trimethoprim, which is a very effective antibiotic against E. coli. So here they had trimethoprim in a concentration of 3. They're using MIC units, which is minimum inhibitory concentration. We don't need to get into the details on that. But what's important is that here they had 10 times more trimethoprim, and here 10 more times trimethoprim, and here there's 1,000 times more trimethoprim than at this point. Okay, and they do the same thing on this side, is to have like an inherent replication on the same plate. Right, so the two halves are identical to each other, and so you're watching two replicates. And what's going to happen in the video I'm going to show next is that the E. coli on each side will attempt to colonize more and more of the Petri plate, meaning that they grow more and more towards the center. But in order to make progress towards the center, they have to be able to withstand these concentrations of trimethoprim. And at the beginning of the experiment, they demonstrate that the starting E. coli have no resistance whatsoever to trimethoprim. They die immediately at certainly at this unit of 3, and they certainly can't withstand any of the higher concentrations at the beginning of the experiment. So if you do see any E. coli growing towards the center of the plate, it means that there are new mutations arising that are selected for during this one experiment that we are going to watch right now. Okay, so here it goes. So here are the two sides of the Petri plate. These are the starting concentrations of E. coli. You see that they run up against a wall on each side for just a moment. And I believe this video runs over the course of about 12 hours or 24 hours or something like that, maybe a few days. So it's many hours, maybe a couple of days, I forget, sorry, we can look it up in the paper. But still pretty fast. So now on this side, they've already broken through the second wall, so they're already at a concentrations 10 times greater than the first one. On this side now they've broken through the second wall, and you can see, I'm going to pause it now, on this side especially, you can see that the new populations growing are descended from very narrow bands that you could imagine all descended from a single colony, or ultimately a single cell. So you can actually see in real time these huge blooms of population all over the place that are evolving from a single distinct ancestor, right? So each one of these blooms from a separate lineage are independent evolutionary events, independent mutations that have been selected for by these conditions. Okay. You can also see some contaminants. So there's perhaps a fungal spore that has landed on the Petri plate and started growing. So it's slightly embarrassing for them. Third wall, this side is broken through. And now we are perhaps seeing perhaps slightly fewer populations that are making it all the way through. It doesn't seem quite so crowded anymore, but they're certainly taking up all of the available space. Here it's even slower, right, and even more sparse. It's not developing quite so fast here in the very middle, where it's 1,000 times more concentrated here than in the center. I think this got taken over by some contaminants, if I'm not mistaken. But here on this side, you can see this really beautiful biologically evolution taking place. And if these shapes are calling to mind phylogenetic trees, because that's exactly what they are. So in this figure, they trace the phylogeny of the populations that made it all the way to the center. So they labeled each of these blobs in the center that made it all the way that far, and then trace, just by observing them, really just looking at this video very, very carefully, tracing back where they came from. And this is basically tracing a phylogeny, a phylogenetic tree that was constructed in real time under this camera over the course of, I think it was a couple of days. Sorry, I didn't confirm that. And of course, they're only tracing a few examples, right? They couldn't have done all of them, or it would be a complete mess, right? So these are only a few examples that they've highlighted with their phylogeny. Okay, so again, these are all independent mutations, arose independently, selected upon by selection by the constraint that in order to grow towards the middle, they had to survive these additional concentrations of antibiotics. So here, even without the concept of gene transfer, right, there may be gene transfer going on here, but it's not required to explain what's going on. Just with traditional first-year biology textbook mutation plus natural selection plus rapid generations from E. coli, you can get evolution of novel resistance that was not there before, and a really, really impressive resistance as well, right? 1,000 times greater than the starting concentration. Okay, so there's actually no new biology here. All of this biology was already known, but it was a really beautiful demonstration of a concept, so it's great for teaching. Some of the details of this are interesting and are new and are why it was in a research paper, and you can read about those details here. Those details being exactly how these mutations occurred, which mutations they are, and which genes, what phenotype those mutations conferred, and also, even more interestingly, which mutations gave rise to the final mutations that you find in the final populations, right? And it's one can never predict how a single mutation here that conferred maybe a tiny bit of resistance eventually evolved into different mutations that enabled greater and greater resistances. So that was the research purpose of this, is to figure out how mutations give rise to new mutations. But the simple idea that E. coli can evolve novel resistances, that was already known. Okay, so that's an example for your writing assignments of how you can identify what was known and what was not known. And it's often a pretty subtle a difference between what was found in the research paper versus what was known before, because that's how science works in tiny baby steps. Okay, so a little bit more about just the basic biology of what's going on and why resistance is such an inherent problem of biology. So here's my simplistic drawing of a population of bacteria. They are producing an antibiotic to kill their enemies. And this only works if they have a defense against their own antibiotic, right? Bacteria live in a world governed by diffusion, right? So if they are making a molecule, they have to be resistant to their own molecule or they are committing suicide. So that simple idea right there is the reason why there will always be resistance to antibiotics. There's no such thing as an antibiotic that cannot that can never have resistance evolved against it, because in order for an antibiotic to be invented by a bacterial species in the first place, that bacterial species must have already invented the resistance mechanism to the antibiotic, or else it wouldn't have ever been able to survive the invention of the antibiotic in the first place. Okay, there's some research by chemists to invent antibiotics, so human-invented antibiotics that are theoretically resistance can't evolve in response to it, and there are some studies on that. I don't know that any of them have actually been verified in real-world studies, but that's the only way you could possibly do it, is if a human invents some chemical that for some reason resistance can't evolve to it. Because any bacterially produced antibiotic has an inherent resistance against it. Okay, and then once a technology has been invented, humans can't control where that technology goes, and bacteria can't control where that technology goes either. That resistance gets passed around to other members of the community. It can get passed around from themselves, but also their enemies, for example, can evolve independent mutations against the antibiotic, as we just saw in those E. coli, and then those enemies can also be the source of sharing those genes to other members of the community, okay? Because once this antibiotic has been invented, there's a very strong incentive for everybody to evolve a resistance against it. So in this cartoon, I'm just stepping through the basic logic of once there's a single mutation, as indicated by the green asterisk, against an antibiotic, there's a very strong selection pressure for anybody with that mutation to do really well. So as indicated by this population blooming, and if that gene gets passed around, with that particular mutation, then other species can also do really well in this environment, which is impacted by this antibiotic. Okay, so I've already mentioned this as one of the big challenges of antibiotic resistance, is that not only can bacteria evolve independent mutations spontaneously against a new antibiotic that they've never seen before, in addition to that, they can also share those successful mutations with completely unrelated species via horizontal gene transfer. And that's how it goes so fast, even faster than what we saw in that video. Because again, that video did not require gene transfer. So it's not only fast, but also spreads among completely unrelated species. Okay, so now just a little bit about the biology of horizontal gene transfer. It's a really important concept in all of microbiology. Antibiotic resistance is only one example of the importance of horizontal gene transfer. It's so important that people think about it and argue about it in terms of the whole tree of life, and whether we can even correctly think about the tree of life as a tree. Because if genes are being transferred among different branches of the tree rampantly, so here there are just two examples involving the cyanobacteria becoming chloroplasts and the proteobacteria becoming mitochondria. Those are two fundamental examples of horizontal gene transfer. But if there's lots and lots and lots of horizontal gene transfer among all three domains of life, then it doesn't really look like a tree anymore. Maybe we would more correctly think about the diversity of life on Earth as a web of life. So here's a very famous figure from a while ago now introducing this concept, and people still talk about this and argue about this today. So it's a fun thing to think about. Of course, we still make trees because phylogenetic trees are very, very useful. All right, so a little bit of teaching here. 3 different mechanisms for horizontal gene transfer I want you to be aware of. One is conjugation, colloquially known as bacterial sex, because it requires cell-to-cell contact. So you kind of get the idea why this is called bacterial sex. You kind of have two parents. They make physical contact with each other. They exchange genetic information through this structure that is usually called a pilus. More than one pilus is called a pili. The transferred DNA is usually in the form of plasmids. So it's usually circular bits of DNA, not the main chromosome that is being transferred during conjugation. So usually it's plasmids that are transferred during conjugation. And this is a form of sexual recombination between species because the plasmids can actually be incorporated into the genome. And usually conjugation occurs much more often within a single species or between very similar species, but it has also been shown to occur between different species as well. So, and here's just the textbook diagram of exactly how this works, with the plasmid being transferred through the pilus, unrolling and inserting to the donor cell, from the donor cell to the new cell. In case you want a little more information about how that works, but in this course I generally don't go in too deep into the molecular mechanisms. Gross example of conjugation is MRSA, methicillin-resistant staphylococcus. Here's an unfortunate individual with a MRSA infection. Methicillin, it's called methicillin-resistant because methicillin was kind of the last resort antibiotic that was successful against staphylococcus. And so once it became resistant to methicillin, we kind of ran out of ideas. And that's why the infection can get to that point, is because there isn't much after methicillin. You can read about papers that are other strategies that are often but not always successful against MRSA. It can be a deadly infection. But yeah, that's why it's called methicillin-resistant staphylococcus. When does this happen? So in a more basic ecological way, why do cells exchange DNA? In general, you can think of it as a stress response, and exposure to antibiotics is one very specific kind of stress, but it happens generally with lots of different kinds of stress. And so here's an image I really like from an old study, where here's a microscope image of a population of cells in a biofilm. That biofilm is just a surface-attached community, so instead of being dispersed in water, they're living on a surface. And they have lots of nutrients, they're very happy, nothing much is going on. And then in this image, they've starved this population of cells. So the first thing you can see is that they're clumped together. So that is a very common reaction among microbes in general, is that when they are stressed, they clump together. So they're in very close contact to other cells. Okay, and then in this study, they were able to use different colors of probes different color labels in their microscope study to label cells that have received DNA from a donor. Okay, so cells that are yellow or green have received DNA from a different cell. So this shows that the clumped cells are much more likely to engage in horizontal gene transfer than the cells that are off by themselves. Kind of obvious, you might think, but the basic idea here that horizontal gene transfer most often occurs in cells that are clumped together is a really important concept, because it means that surface attached communities that are very dense, where cells are in very close contact with each other, are very likely hotspots for horizontal gene transfer. So for example, in a wastewater treatment plant, you have big vats of liquid, and you also have surfaces where you get gunky biofilms growing. Those gunky biofilms are probably where most of the horizontal gene transfer is occurring, not in the big vats of liquid. Okay, so that's a practical application of a really basic biological result. Second, or one of the other methods of gene transfer is transduction, which is just a fancy word for DNA that has been transferred by viruses. We're going to talk about viruses later in the course, but there are lots and lots of viruses in the world. It's been estimated that there are 10 million viruses in every milliliter of seawater, so that's like one drop of seawater has 10 million viruses, because bacteria are constantly being infected by viruses. We often think about viruses as things that cause illness in animals, but bacteria were the first, bacteria and archaea were the first hosts of viruses, and they're still the bacterial viruses are still the most common viruses in the world. And someone estimated that if you stack all of the viruses in the ocean end to end, they would stretch past not only to the end of our own galaxy or to the next galaxy, but past the next 60 galaxies. That's just a crazy number. Another way of putting it is that there are 10 to the 23, so 23 zeros, viral infections every second in the ocean. So bacteria and archaea and microbial eukaryotes are infected by viruses that ridiculously large number of times every single second, because there are so many microbes and there are so many viruses, and you estimate how often they get infected, and that's what you get. These are just crazy numbers to try to wrap our minds around the fact that there are lots of viruses in the world. Very, 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 very few of them have anything to do with humans. So those are the three mechanisms for horizontal gene transfer. I didn't mention transformation, which is basically the same as conjugation, except that there's no physical contact. So there is a DNA molecule just kind of floating around the environment, and a cell grabs it through their cell wall and through their cell membrane. And again, this almost always happens as a stress response. The cell is starved, it's about to die, it's doing last resort strategies of anything that might help it survive. And so one of those strategies is to grab random pieces of DNA that are floating along, right? This is a last resort, and maybe that piece of DNA is the thing that it needs. So this is perhaps more important we really know this because of lab experiments. It's a useful tool to use in the lab. It's not clear how useful it is in nature, but it has been demonstrated to occur. For example, biofilm formation can be triggered by the presence of free-floating DNA. So cells can, at the very least, certainly recognize when there is DNA floating around in their environment, which is a pretty interesting thing to know. All right, so that was some basic biology, getting back to antibiotic resistance in particular, and I'm going to finish with some human-oriented practical things and ways to think about this as a practical and urgent problem that we need to solve. So this slide is showing the many different ways that antibiotic resistance can spread in our human systems. So not talking about different molecular mechanisms of DNA, but different human systems and how humans are causing this to occur and making it worse more than it would be otherwise. So starting on this side of the figure, here's an example of George, who takes antibiotics, and anytime you bring antibiotics into your system, you are inherently creating an opportunity for resistance to evolve. So he's now evolved a new population of resistant bacteria in his gut. He goes to a hospital because he's very sick, and those microbes can be spread to other patients. It can be spread to the healthcare workers. They go out into their community and spread it with each other. And even if he doesn't go to the hospital, he can still spread it with the people he lives with, right? So these are really kind of obvious ideas, but just to point out that it's not just a hospital problem. It's a complex human culture problem. In addition to this human-only set of interactions, we also have the problem of agriculture, because most of the antibiotic usage is actually in agriculture, not in human medicine. So I forget what the number is, but the vast majority of all the antibiotic usage in the world, or at least in Western countries, is with livestock. I'm pretty sure it's within the world, actually. Because there's so many antibiotics being used, it's a really great way to develop resistance, especially because most antibiotics are not being used to treat any illness. They're used because it just turns out that if you give animals antibiotics, they get really big and really fat really fast. So that it's not being used to treat a disease. So you get drug-resistant bacteria really easily, and when you eat that meat, then that is one potential mechanism for spreading. But perhaps probably more importantly is the runoff. So fertilizer and contaminated water that gets into the water table, runs off into our water systems, or it gets onto crops on a farm that then get harvested and then sold in the grocery store. These are mechanisms that have been demonstrated to be ways that people get exposed to drug-resistant bacteria. So point of the slide is it's a complex problem. There are many, many different ways that we are making the problem worse, and it's not something that can be solved with just one fix in one area. It's a policy problem as much as a scientific problem. And this is the same slide. Basically, again, there's the hospital component to it. There's an environmental, agricultural component to it. And this is really just a way of recommending this review, which talks about how in order to make progress with this, we not only need scientific research, we not only need to discover new antibiotics, which is of course necessary, but just as important and arguably more important is to develop good policy frameworks and environmental policies and hospital policies and pharmaceutical policies that can at least give us a chance to keep this under control. All right, I'm sure we'll talk about this more in class, and so I will stop talking now and highly recommend this review in particular and the others that I've already posted on Canvas. See you later.